So what are proteins and what are polypeptides? How do they differ, and what are the composition of the proteins, how they are made up? These things we are going to discuss. And what happens when a protein is digested, means it forms further the shorter type of molecules. So all this we are going to cover in this video. So let us come to protein. What are protein? When we discuss about protein, the one thing that one must remember is that the protein is the building block of our body. Protein is the building block of our body, but they itself made up of amino acids. Means we can say, it's like the city is made up of houses. The city is made up of houses where the house can represent the protein, but the protein is itself made up of bricks, or the protein is itself made up of amino acid. The house is itself made up of bricks, and that brick is the amino acid. So protein is actually made up of, they are made up of amino acids. Amino acids. So in a simple sense we can say that when we talk about protein, so we first need to talk about amino acids. These are the amino acids which connect together in this fashion. These are the amino acids, the building block of the protein, that connect with each other in this fashion, and they form a long chain, and that long chain is known as polypeptide, Polypeptide is nothing but the raw building material, of the protein. That is the polypeptide. That polypeptide is formed by the combination of amino acids. Now in a protein, there could be more than one polypeptide. Remember, the one chain of amino acid is known as polypeptide. And generally it contains more than 50 amino acids. Okay, but in a protein there could be multiple polypeptides, or you can say that if this is the one chain of polypeptide, means one amino acid chain, this could be the another amino acid chain. So it is the one polypeptide, it is another polypeptide, but they together form the protein. In a protein there may be single polypeptide, or there may be multiple polypeptide depending upon what type of protein it is. Like we can take the example of hemoglobin. Hemoglobin is made up of 4 polypeptide chain. Means these are the chain, this is similar to this. It is a straight chain, but when it folds upon itself, it seems like this. So hemoglobin is itself made up of 4 polypeptide chain, and we name them alpha 1, alpha 2, beta 1 and beta 2. So it is alpha 1, alpha 2 and beta 1, beta 2. Means we have given the name to these polypeptide chains. This is alpha 1, alpha 2, beta 1 and beta 2. So I am summarizing. What are proteins? Proteins, they are the main building block of our body. They are the main performer of our body. They perform all kind of function in our body. That is the proteins. Protein, it's itself made up of amino acid. When amino acids join together, they form a long chain known as polypeptide. In a protein, there could be one polypeptide chain, or there could be multiple polypeptide chains. Altogether, the set of the polypeptide chain, if there are more than one, they form the protein. So this is the way the protein is present. Now, if we come to this, that how these amino acids join together and how do they form protein. So let us take an example of an amino acid first. As the name suggests that it is amino acid. Amino acid means it must be having a amine group and it must be having a acidic group, where the amine group we represent as NH2, where the amine group we represent as NH2. This is the amine group, and when we talk about the acidic group, then it is the COOH, carboxyl group. This is the. So amino acid would be having a NH2, a amine group, and a carboxylic group, or the acidic group. Now, because these two groups are opposite in nature. This is basic in nature, this is basic in nature, and this is acidic in nature. So altogether amino acid is having a acidic group and a basic group. They are attached. They attach to the common carbon, that is, this is the common carbon with which this carboxylic group is attached, and on the other hand this amine group is attached. But to satisfy the valency of the carbon, we must need to place a hydrogen and we must need to place another group, that is R group. Now what is this R group? This is a variable group. Variable group means in one particular amino acid the R group could be hydrogen. In some other amino acid the R group could be CH3. In some other amino acid the R group could be CH2OH. Means it is that kind of group that vary from amino acid to amino acid. So depending upon what type of amino acid it is, this R group vary. For the time being, to keep it simplified, to keep it simple, I am assuming that there is a R group. Now because this is having acidic character, and this is having a basic character, this amine group is basic in nature and this COH group is acidic in nature, both almost neutralized to each other. Now the main impact is arise because of this R group. Now this R group could be of acidic, basic or neutral nature. According to that we can define the nature of the amino acid. Means the amino acid nature, whether it could be acidic, basic, alcoholic or so on, is dependent mainly upon this R group, mainly upon this R group. Okay, now understand this thing. There are two carbons. If we exclude the carbon if present in this R group, there are two carbons. That is, this is the alpha carbon and this is the beta carbon. This is the alpha carbon and this is beta carbon. Now because all the functional groups like amine group, this hydrogen, this is COH and this R group is attached with the alpha carbon. So we do say here that these amino acid are termed as alpha amino acid. They are termed as alpha amino acid because all the functional group are attached with the alpha carbon. So you got it. Now depending upon what type of the R group it could be, we can define the nature of the amino acid. But before going into that, amino acid can also be defined in some other way, like they could be essential or non-essential, or they are like polar amino acid or non-polar amino acid, depending upon their what they are, whether they are charged or they are uncharged at a particular pH. So that we will discuss later on, but first understand that how the protein are made from these amino acid. Now suppose this is an amino acid. We take an example of the amino acid. Now I am removing this rest of the things. If this is an amino acid, this is an R group that is attached with the alpha carbon. This is an amine group. If I take another amino acid, say suppose if I take this as H, so it is glycine. The name of the amino acid is glycine. And if I take another amino acid, that is CCOOH, here it is H and it is NHH. Just to simplify, I am writing it in this way, that this NHH and here it is CH3. So it is alanine. The name of the amino acid is alanine. So I took two amino acids, the one is the glycine and another is the alanine. The basic difference is only the R group. In glycine, the R group is hydrogen, while in case of alanine, the R group is CH3. Now, how they combine with each other? When they combine with each other, it's a dehydration reaction. It's a dehydration reaction leading to the release of water molecule. It's a dehydration reaction. It's a dehydration reaction here. And this is the H, this is the OH group that is released in the form of the H2O. So after this, we can rewrite this is like it is CO and it is an H and it is COOH, this is H and this is CH3, this is NH3. H2, this is H, and this is H. Now remember, see, this is NH2, this is NH2 here, this is carbon, this is HH, we have placed it, and this is CO that is left remain because OH is removing in the form of water along with this hydrogen. So this is CO and this is NH that left remain. This is left remain, and this is the CH3, this is CaCH3, and this is COOH. So in this manner, this C double bond O, we can write it in this manner also. It's like this. So you see that a kind of bond is formed. This bond is known as peptide bond. This bond is known as peptide bond. Peptide bond connect amino acid together. So we have seen that two amino acids are connected together by this bond. Means if I assume that these are the two amino acids, this is the glycine, this is the glycine, this is the alanine, they connect in such a way that they form the peptide bond. Now remember, always remember, this is the first amino acid because here amine group is free, and this terminal we known as amino terminal. This we call as amino terminal. It is amino terminal, or it is N terminal, or it is N terminal. It is N terminal, this one. And where the carboxylic group is free, suppose if I add this many, so there must be carboxylic group free here, and here it must be the NH2. Here it must be the NH2. So this is the amine group. This is the amine group that is free. This is the amino terminal. And at the end where the COH group free is free, this is known as C terminal. It is known as C terminal or carboxylic terminal, carboxy terminal, sorry, it is carboxy terminal. So this is carboxy terminal. So in this manner the two amino acid attach. Now if further amino acid need to be attached, we need to add to this C terminal. So in this manner you can hundreds or maybe even thousands of amino acid can be attached, but the basic pattern would be the same, like this is the first amino acid, this is the second amino acid, and so on, and gradually will found here, and in between the two amino acid you will find a type of bond that is the peptide bond. So everywhere, here, these are the peptide bond. Between two amino acid there is one peptide bond. Between three amino acid there is two peptide bond, and so on. In this manner, a long polypeptide chain is formed, and then this polypeptide chain assemble in such a way, either singly or in multiple, they form the proteins. So that is what. This is the English version. If you want to see the Hindi version, that is also available. Follow the Gyan Guru and #life. And this video is useful for NEET aspirant as well as those who are preparing for SET examination and various competitive examination related to biology.